thing we are going to learn is the most important thing in the chapter of DNA Core of Life, which is what? DNA replication. All right. So what exactly is DNA replication? So DNA replication is a process by which DNA makes an exact copy of itself. All right. So you should know this definition also, because they will probably ask you this in exam, which will consist of 2 marks. And it's and the definition is very simple, like replicate. Okay, so firstly, what's the meaning of replicate? To copy something. So what's the definition of DNA replication? It's the same. It's the process by which DNA makes an exact copy of itself. So you should know this definition. Okay, so it occurs during the interface of cell division. Sorry, don't mind that. So it occurs during the interface of cell division. So we'll learn more about, or more deep about, the cells during the meiosis lesson. But you should know that it occurs during the interface of cell division, which is the first state of meiosis, you can say that, or cell division. Move into the process of DNA replication. So one thing I know for sure is that you guys really complicate this thing. All right. So I know that it's not that easy to cram, but there is no need for you to cram this thing. If you understand it properly, then it would be easier for you to remember. All right. So let's move. Okay. So DNA double helix unwind. So the DNA has a double helix shape, right? And it's twisted together. So the first thing that happens is the DNA double helix unwind. All right. Then the weak hydrogen bonds between the nitrogenous bases are broken. So we know that the nitrogenous bases are attached to each other by weak hydrogen bonds. So they basically break, right? And then the DNA stands separate. So basically they unzip, right? So they unzip. All right, so each original DNA strand serves as a template. So you might be wondering, what is template? So this template is one strand of DNA that is used as a guide to build another one. All right, so each original DNA strand serves as a template on which its complement is built. All right, so let's say this is the original DNA strand. So I'm just explaining what the template means. So the original strand acts as a guide to build the another strand, the complementary strand. Okay, so then the free nucleotides build a DNA strand onto, okay, wait, I can't really, all right. Okay, so the free nucleotides build a DNA strand onto each of the original DNA strand. All right, so the free nucleotides, okay, so the free nucleotides build a DNA strand onto each of the original strand, attaching their complementary bases. All right, which means adenine will go to thymine and cytosine would go to guanine. That is basically what's happening here. All right. Then so this results in two identical DNA molecules. Each molecule consists of one original strand and then one new strand. All right, so this might be original, and then this might be, and then that might be a new strand. So that results in two identical DNA molecules. All right, so let me give you a simpler version that you can memorize for the exam. All right, so this is exactly the same that I explained. So you should just take a screenshot, breaking that down here. So this one is the simplified version. So you can just take a screenshot of it, and then you can memorize it. This will consist of 7 marks in your exam. So let's just revise it one more time. So the DNA double helix unwind. The weak hydrogen bonds between the nitrogenous bases are broken and the DNA strands separate. each original strand serves as a template or as a guide for building its complement. Then free nucleotides build a DNA strand onto each of the original DNA strand, attaching complementary nitrogenous bases. And this results into identical DNA molecule, each consisting of one original strand and one new strand. Alright, so you should just memorize this for your exam. So now we should know what's the importance of DNA replication, and you should know this. I'm telling you, this will definitely come in your exam. Let's say it might come into your first term exam, and if not, it will definitely come to your trials or your final exam. So you should know it. All right. So what's the importance of DNA replication? So DNA replication is important for cell division, particularly mitosis. Then it allows each chromosome to be copied so that new identical daughter cell produced contains the same number and type of chromosomes. All right, so you should know this because this will definitely come in your exam. Okay. Then what are the errors that occur during DNA replication? All right. So errors that occur during DNA replication may sometimes lead to mutation. Now you might be wondering what is mutation. So mutation is a change in nitrogenous base sequence. Okay. So what is the change in nitrogenous base sequence? Perhaps adenine might go with cytosine, then guanine might go with thymine. So this is a change in nitrogenous base sequence. All right. So if the incorrect nitrogenous base is attached to the original strand and a nitrogenous base is added or deleted, this might lead to the sequence or order of bases changes on the DNA new molecule and resulting in a change in gene structure. Okay. When the gene structure or the nitrogenous base sequence changes, it might lead to diseases. Okay. Then I have this practice question over here. So you can try to attempt this practice question. Okay. So let's start answering it. So, all right. Name the process illustrated in the diagram above. So what's this process? What is this process? So this process is DNA replication. Now you all might be wondering, how do you know that this process here is DNA replication? So here you can see that the DNA is twisted together, but then here it basically unwinds, which means that when the DNA double helix unwind, or that means that DNA replication is taking place. So the answer for here is DNA replication. Okay. Then state the significance of the process mentioned in question 1. So what's the importance? All right. So the importance is that to produce two identical DNA molecules with the same number of chromosomes. Okay. All right. Let's look into number 3. So number 3 is saying that identify the parts labeled 1, 2, and 3. So here 1. So I think number 1 we all got it correct, which is deoxyribose sugar. Okay. Then number 2. So here number 2, I think most of you said it's weak hydrogen bonds. No guys, they are not asking that. All right. So here, let's identify it first. So this one is a deoxyribose sugar. We already said that. Then this one is a nitrogenous base. So what might this be? That is a phosphate. So that is a phosphate. So it's P out there. Number 3, we all are clear. It's a nucleotide. All right. So number 4, we know that this one is a nitrogenous base. But we can't simply say that it's a nitrogenous base. We have to identify it. All right. So now we are wondering how would we identify that? So as you can see here, we have adenine here. What goes with adenine? Thymine. So what would that be? So what would number 4 be? Number 4 is basically thymine. And yeah, so if they would ask you what's this, then you would simply say that's so that's a cytosine. But then here they're asking for this, so that would be thymine. Okay. Then the number 4, they are asking us describe how this process takes place. So they're basically asking you for the process of DNA replication. And see, it consists of 6 marks. I think here is 6 marks, but in exam that would be 7. So that's your free 7 marks. So what you simply do, alright, so what you simply do is that you just write this entire process and then you'll get your 6 to 7 mark. Okay, then alright, let's move on. Alright, they're asking us give one location of extranuclear DNA. Okay, so we discussed about this in our previous lesson. So the process, okay, so the extranuclear DNA is found in the mitochondria, known as mitochondrial DNA, and it's found in the chloroplast, known as chloroplastic DNA. So yeah, there are the two location, but here they are only asking you for one, so you can write one of them.